Namaskar. In a second class, we will discuss about the structure, the anatomical structure of the human testis, and what are the basic structure present in seminiferous tubule. What are the different parts of the seminiferous tubules? So let's start. This is a second part of our first class, the human reproduction. This is a human testis, LS, longitudinal section, and if we carefully notice, normally in a NEET exams, it is questions are asked from several points of the testis. A schematic diagram will be given, and you will be asked the answer, the different point. What are the different points? Suppose it will be mentioned that A, what is B, what is C, and option will be there. So let's start. What is actually what? This is a longitudinal section of testis. Testis is actually full of seminiferous tubules, highly convoluted seminiferous tubules. Convoluted means highly spirally coiled. Seminiferous tubules, these are the seminiferous tubules, and the outer layer of the testis. Outer layer of testis is called tunica vaginalis. Tunica vaginalis. Tunica vaginalis terms is also present in a ovary structure also. So this is a tunica vaginalis, and just below the tunica vaginalis there is a tunica albuginea. Tunica albuginea. This is a tunica. Tunica albuginea is present here. Tunica vaginalis is just outer layer. So tunica vaginalis is inserted or invaginates inside, folded inside. Tunica vaginalis, tunica albuginea, folded inside and in a finger-like projections to form lobus testis. That is, this is a tunica albuginea, invaginates inside the testis, testis, and this is called scrotum. Actually, scrotum means a sac-like structure within which the testis is present. Scrotum layer is present outside here. This is not a scrotum. Scrotum is actually present in outside. This is a skin or sac-like structure. This is a scrotal layer. So below the scrotal layer, this is tunica albuginea. Tunica albuginea invaginates finger-like projections inside and form chambers, small chambers. These chambers are called lobus testis. Inside the lobus testis, highly coiled seminiferous tubules are present. Highly coiled seminiferous tubules. These seminiferous tubules is present in a highly coiled condition. Because why it is present in such convoluted condition, highly coiled condition? Because huge is a very long sized seminiferous tubule is need to attach or need to pack inside the testis. So why to produce huge amount of sperms, it is needed that seminiferous tubules will also be very long. So in a small place, how a long seminiferous tubule will be adjusted by convolution, huge amount of folding. However, this is called lobus testis, and these convoluted tubules they forms a straight structure. When they comes together, and these convoluted structures together release from the testis here, release from the testis to a secondary structure here, also a long secondary structure. This is called epididymis. So the convoluted tubules, forms tubuli recti. This convoluted tubule, that is the seminiferous tubules, they forms a straight and somewhat straight seminiferous tubule region. There is a red mark here. This is called tubuli recti. Tubuli recti is present between. It is actually a sandwich condition. It is present just between the seminiferous tubule and this one is retitestis. Reti means reticulate. That is, seminiferous tubules are highly reticulate structure. In this region, seminiferous tubules forms the reticulate structure. This is called retitestis. So the tubuli recti present between the retitestis and seminiferous tubules, highly convoluted seminiferous tubules. So this is the tubuli recti, and then tubuli recti, the retitestis and retitestis together fused again, and from the retitestis the seminiferous tubules together release outside the testis and form a highly coiled structure here also called ductus efferentia. Ductus efferentia also known as vas efferentia. Vas efferentia. Another close term is present, vas deferens. Vas deferens and vas efferentia is two different things. Here vas efferentia is present just outside the retitestis, that is reticulated seminiferous tubules. Reticulated seminiferous tubules together form a structure called vas efferentia or ductus efferentia. Then from ductus efferentia, another secondary structure is present, is called epididymis. Epididymis is very vital. We know that in a previous section we have discussed that epididymis has three parts. There is the head region, there is the middle region and lower region. Head region is called caput, middle region is called corpus and tail region is called cauda. So what is the function of epididymis? The secondary structure. This secondary structure, why it is present? As we mentioned that inside the seminiferous tubule, here the seminiferous tubule, we have section here, a single seminiferous tubule, two seminiferous tubule here. It is the frontal section of seminiferous tubule. What is actually the seminiferous tubule? I am discussing now. Inside the seminiferous tubule, you know that sperms are developed, but sperms are present in immature condition inside the seminiferous tubule. Inside the seminiferous tubule, sperms are immature. So in the epididymis, from upper, middle and lower parts, gradually it is moving from upper to lower part and sperms getting mature, so and motile. So it is a different part: caput epididymis, corpus epididymis and cauda epididymis. However, after the epididymis, another region is present, tubular region is present, it's called vas deferens. What is actually the vas deferens? Vas deferens is a storage site of sperms. Sperms are developed inside the seminiferous tubules and they are moving from here, from tubuli recti to retitestis to vas efferentia to epididymis to vas deferens. So this is a region. But first of all, seminiferous tubule. This is a seminiferous tubule. From seminiferous tubule, gradually it is called tubuli recti. Tubuli recti. From tubuli recti, so retitestis. Retitestis. Retitestis to vas efferentia. Vas efferentia. Not differentia. Efferentia. Vas efferentia. From vas efferentia, it is called epididymis. Epididymis. From epididymis to vas deferens. From vas deferens to ejaculatory duct. Ejaculatory duct. So this is the overall structure of a testis. Now, if we do a cross section of a seminiferous tubule, we will see that a seminiferous tubule is a tube-like structure, and this outer layer is called germinal epithelium layer. This is a germinal epithelium layer, and this from the germinal epithelium layer, the large cells, these are called spermatogonium. Here, the first layer just below the germinal epithelium is called spermatogonium. From spermatogonium, as you know that by mitosis division, from the primary spermatocyte. So just below the layer is called primary spermatocyte. This is spermatogonium, this is the primary spermatocyte, and below the primary spermatocyte, secondary spermatocyte, and then spermatids. So if we look a close look here, what will actually happen, where the actually meiosis occur, and what is actually the diploid and haploid number of cells. Here the spermatogonium, first cell. Spermatogonium undergo mitosis to form two spermatogonium again. One spermatogonium will back, that is, it is present as a stored spermatogonium, and second spermatogonium will again divide in a mitotically to produce two primary spermatocyte. So from spermatogonium to primary spermatocyte, it is a mitosis division. So up to the primary spermatocyte, it is diploid. Cell is diploid. Then after primary spermatocytes to secondary spermatocyte, first meiotic division occur. So first meiotic division occur between primary and secondary spermatocytes. To develop the secondary spermatocytes, first meiotic division is needed. Then from secondary spermatocytes, then spermatids are developed by meiotic 2. So this is the underlying region. These two underlying regions are the meiosis 1 and meiosis 2. So you know that here from one cell 4 spermatids are developed. From 4 spermatids, after differentiation, the sperms will develop. So it is very important. Questions are asking from here. This is a cross section of seminiferous tubule, and what is actually these long cells? These are the Sertoli cells. Sertoli cells are the nourishment providing cells. Nourishment providing cells present inside the seminiferous tubule. These Sertoli cells are highly sensitive to FSH, follicle stimulating hormone. So here is a regulatory pathway present. FSH and LH how regulate the production and nourishment and control the production of its sperms. In this case, in between the two seminiferous tubule, outside a cells are present, a bunch of cells are present. These are called Leydig cells. Leydig cells are very important for the production of testosterone or androgens. Androgens are responsible for the production of secondary characters, secondary sex organs of the male body. So these are the Leydig cells. Leydig cells are highly sensitive to luteinizing hormone. Both FSH and LH are produced by the anterior pituitary hormone. So this is very important. So here is the regulatory point. Please closely look here. This is the hypothalamus. We know that a hormone, all hormones, then mainly the release factor hormones are produced by the hypothalamus. So gonadotropic releasing hormone, gonadotropic release factor hormones are produced by the hypothalamus. They mix to the blood and come to the anterior pituitary. Anterior pituitary, by getting a signal from gonadotropic releasing hormone, they produce two hormones, FSH and LH. Follicle stimulating hormone and luteinizing hormone. Follicle stimulating luteinizing hormones are also known as ICSH in case of male. We know that interstitial cell stimulating hormone, ICSH, interstitial cell stimulating hormone. Now, what will happen? FSH target is Sertoli cell. FSH target is Sertoli cell. It activates the Sertoli cell. Sertoli cell will produce Sertoli cell. What will Sertoli cell occur? Sertoli cell helps in spermatogenesis, as well as Sertoli cell will develop inhibin. Inhibin is another hormone. That inhibin, if sufficient amount of production is occurred, that is sufficient amount of spermatogenesis occurred in a normal way, then or more than sufficient spermatogenesis occur, then inhibin will form a negative feedback loop by inhibiting anterior pituitary. Here inhibin is high, inhibit the anterior pituitary, and LH target the Leydig cells. Leydig cells produce the testosterone. Testosterone has an important role in spermatogenesis. If sufficient or more than sufficient spermatogenesis is occurred, then testosterone will go in a two place. First, anterior pituitary, they will inhibit anterior pituitary, that is no need of LH, and then inhibit the hypothalamus, that is gonadotropic releasing hormone should be stopped. So this is the regulation, negative feedback regulation present in this case. So we have to remember this, the different cell, how the primary and secondary spermatocytes are developed, how spermatids are developed, and what are the different steps from seminiferous tubules to ejaculatory duct, how the sperms are moving. And this is the secondary storehouse of vas deferens. Vas deferens is secondary storehouse of that is the storehouse of sperms. So this is the tunica albuginea, tunica vaginalis, and this is the overall structure of a testis and seminiferous tubule. Thank you.